So finally, we get to these two men named Watson and Crick. And like I said before, we're leaving out part of the story because there are other people's names that should go into the mix as well that get shunned. But Watson and Crick is important. It seems like every time we talk about DNA, we look at DNA and we think Watson and Crick because this is what we're told in our biology classes and in our science courses. Well, Watson and Crick does have an importance. What do they do? Well, they determine the structure of DNA. They determine the structure and 3D shape of DNA, and that is why they became so important. So, more about them later. We're actually not going to delve really into their stories yet because we need to start talking about the individual pieces and parts that make DNA up before we go further. So, we can't understand really Watson and Crick's discovery until we go on and talk about chemical structure, at least, of the pieces that make DNA up. And that's exactly what we're going to do. So, with the nucleic acid world, the DNA world, we need to take a look at the beginnings. Give it to me in baby steps, right? That's what we always really want. So, everything up to this point has been about polymers. We've talked about sugars hooking up together to make polysaccharides. We talked about amino acids hooking up together to make proteins. So we're going to have something that hooks up together to make DNA. So DNA is a polymer. That's the first thing that you need to realize. Again, polymer means many unit. DNA is made up of many units. What are those units? Well, there's a couple of terms for those depending on what piece and what part of the molecule that you're talking about. So, in the world of DNA, there are three versions of this structure that we need to make sure that we understand. So, the first one is something called a nucleic acid. And then we can talk about a nucleoside, and then we can talk about a nucleotide. So very similar in names, and that's what gives people a lot of problems because there's only a one letter difference in two of them. Nucleoside and nucleotide, they are different. So these are the individual components that will hook up together to give us this deoxyribonucleic acid, otherwise known as DNA. So DNA is something that we call a macromolecule. Macro means big. So that means it's fairly large. And we can kind of see that because, like I said before, I can go and I can take a look at a video that shows extracting DNA out of a strawberry, and I can see the DNA from the bag as I wind it on the actual spindle or the glass stirring rod. I can see it. Polysaccharides I can't really see sometimes, but DNA I can. So we call that macro, M-A-C-R-O, not micro of M-I-C-R-O. So these are fairly large compounds that I'm dealing with. Okay, so what's the difference here in the names? Well, a nucleotide is made up of a nucleoside, and the nucleosides have a phosphate group. The nucleosides are composed of an aldopentose sugar linked to a nitrogen of a purine or pyrimidine base. Okay, so let me read that again because I think there's some terms here that we know now and we can kind of maybe realize why they were so important back in the day. A nucleoside is composed of an aldopentose sugar. Aldo is the aldehyde. Pentose is a 5-carbon chain, and this 5-carbon chain that has the aldehyde functional group, I can kind of imagine what that looks like, right? A COH on the end, and 4 more carbons attached, each with an OH group on them. So I'm starting with an aldopentose, and I hook that up to a nitrogen. And that nitrogen is involved in a purine or pyrimidine base. And purine and pyrimidine are bases that we've talked about in organic chemistry already. So these are structures you might not remember, but these are structures that you have seen before, and we will take a look at the template for these structures and talk about them in more detail. But that is the nucleoside. So the nucleosides hook up to make nucleotides through a phosphate group. All right, so down at the bottom, it says nucleic acids carry genetic information, and there's two common types of nucleic acids. And these nucleic acids are something called DNA and RNA, and these are found in all living things. If it breathes, if it eats, if it sneezes, if it talks, it's got DNA in it. Okay, so all living things, if it grows like a strawberry, it has DNA. Okay, so let's take a look at the nucleic acids. Nucleic acids can form a polymer. Nucleotides are the pieces of nucleic acids. And the nucleotides are composed of a nitrogenous heterocyclic base, a pentose sugar, and a phosphate group. Okay, so fancy words. Nitrogenous basically means nitrogen containing. Heterocyclic means a ring is involved. That's not all carbon. There's something else different in there. A pentose sugar, 5-carbon saccharide, and a phosphate group. So maybe PO4 I'm looking at. We'll see. But this is what the nucleotides are composed of. All right. So we're going to start off simple. And we're first going to start off with these heterocyclic bases. And here is our first heterocyclic base that I need to memorize. I'll say that again. Memorize. So in organic chemistry, I've told you very often, don't memorize stuff, right? If somebody tells you to memorize something, that's bad news. But this structure, I have to kind of memorize to make sure I understand how everything else is falling through. And this is a heterocyclic base, meaning it's not all carbon here, right? I mean, I see some nitrogen. This is a nitrogen there, not a carbon. And that's because it's hetero, which means it's different in the ring. So this is our heterocyclic ring here. These rings typically consist of nitrogen, oxygen, and sulfur. That is what we see in the ring systems. If it's not all carbon and something else has to be there, it's one of those three culprits. And in the world of DNA, it's going to be nitrogen here in every case. So this is a heterocyclic ring. We call it a base because bases accept protons. And this molecule can accept a proton, and this proton acceptor is the nitrogen that's here. This has a free pair of electrons on it. It's got some negatives around it. It has a double bond on it. So this is a base by nature. So we've talked about this structure before. This structure is called pyridine. And if you go back to the alcohol reactions, you are going to see the incorporation of the pyridine for a purpose, and that was a hydrogen scavenger. What it did is that it went through solution, and as this reaction generated hydrogen, this pyridine would take that extra hydrogen and basically use it up. It would kind of preoccupy it so it wouldn't go out and react again with maybe a reagent or a product that we would want. So pyridine is not anything new. We've seen the structure before. We've used this in reactions before. This was kind of like a solvent at some times that we have used, but this is not new. So this is our first heterocyclic base that we call pyridine. All right, so we have another one, and this other base is called purine. And purine is a 6-membered ring, and it is a 5-membered ring that get hooked up side by side. Okay, so with the 6-membered ring, I see 2 nitrogens here, a double bond and a double bond. And then over here with the 5-membered ring, I see 2 double bonds, one here and one there. And I see 2 nitrogens, one here and one there. So this purine you're going to have to memorize as well. These are really the starting pieces for my DNA. So I have the purine base and I have the pyrimidine base, and this is the bottom molecule. So if I go back to this slide, this is pyridine, right? So this structure you have been presented with already. Very similar to the pyridine is a pyrimidine, and this has an extra nitrogen in it. These two bases are what we will find in DNA, purine and pyrimidine. Okay, so you've seen these types of structures before, like the pyridine in the previous slide. But it's purine and pyrimidine that we are focusing on here with the world of DNA. So these are my 2 common nitrogenous heterocyclic bases. They contain nitrogen, they are rings, they are bases, and they're made up of more than just carbon. So purine and pyrimidine. The way that I kept these straight in undergrad is I looked at this molecule, and this was the simplest out of the 2, but it got the most complicated name, pyrimidine. And this was the most complicated molecule out of the 2, and this got the simplest name of purine. So that could be something that I will ask on a test. One of the things that I could ask would be, okay, here's the structure. Is this purine or pyrimidine? And here's this structure. Is that purine or pyrimidine? Or I could tell you to just draw it straight out. I could say draw the purine and draw the pyrimidine, and you would have to draw that from scratch. So be prepared. Whichever way I ask the question, just be able that you can answer it correctly. That's all that we ask. Okay, so this is our first ingredient to the pie. And that pie that we're making is DNA. So in the next video, we'll take a look at the other components. So now that we have been introduced to my bases, what are the other things that make up DNA? And how should I draw those? And what's the importance of those pieces? So again, stay tuned. If you got nowhere to be, you might as well watch the next one. What else you got to do?